Imagine this. You're trekking through the scorching Sahara Desert, miles and miles from any ocean, and you stumble upon whale bones.
Whale bones.
Yeah.
In the Sahara.
Exactly. Not just a few, but an entire valley just teeming with them. It's a real place, Wadi Hitan, the Valley of Whales.
Uh-huh.
Researchers found these amazing fossils, like Dorodon atrox. That's an extinct whale from like 35 million years ago right there.
Wow. So what was once a sea is now desert.
Precisely.
Yeah.
And that incredible find, it's not just a cool story. It's, a fantastic window into the real story of macroevolution.
Ah, the big picture.
Yeah, we're talking about the really immense sweeping changes in life on Earth, the stuff that happens above the species level. Think about how vertebrates first came onto land, or the massive impact of extinctions, or even how flight evolved. It's this grand view of life's history.
That's exactly what we're digging into today. How these fossils, these ancient clues, tell us not just what lived, but how life itself transformed over, well, billions of years. We'll look at the major forces, the drivers behind the rise and fall of whole groups of organisms, and, maybe surprisingly, how genetics plays into these huge shifts.
So the mission is to understand the broad patterns, the kind of rules of the road for life's long journey.
You got it. By the end of this deep dive, you should have a really clear sense of these patterns, even the unpredictable ones, that have shaped life as we know it.
Okay, let's get into it. So our main window into all this deep time, going back billions of years, is the fossil record, right?
That's our primary evidence, yes.
Most fossils, you find them in sedimentary rock layers, these things called strata. But it's not just bones and rocks, is it? I mean, I've seen pictures of insects perfectly preserved in amber.
Oh, yeah, amber's incredible. Or think about that wolf pup they found frozen up in the Yukon, 50,000 years old, perfectly preserved.
Amazing. So fossils come in all sorts of forms.
They do. And what's key here is, while the fossil record is incomplete, I mean, let's face it, most things don't fossilize. Lots get destroyed, or we just haven't found them yet.
Okay, yeah, that makes sense.
It's still incredibly detailed. It shows us, beyond doubt, that organisms from the past were often wildly different from today's. And crucially, it shows how new groups emerged from existing ones. Remember those Wadi Hitan whales?
With the hind limbs, yeah.
Exactly. The fossils show ancestors with hind limbs. It's this clear link, this transition from land back to sea. It's like finding missing chapters in Earth's, well, autobiography.
So if the record is incomplete, like you said, how can we be so sure about these big stories? How do we actually know how old things are? It's not like rocks have dates stamped on them.
No, not exactly. But we have some really clever methods. First, the order of those rock strata, the layers, gives us relative ages.
Like peeling wallpaper, you know which layer is older.
Pretty much. Layer A came before layer B. But for the actual ages, the calendar dates, we need radiometric dating.
Ah, the radioactive stuff.
Exactly. And this is where it gets amazing. It lets us read Earth's clock with incredible precision, going from just older than to pinpointing dates millions, even billions of years ago.
Okay, how does that work? Sounds complicated.
The basic idea is half-life. You have these radioactive parent isotopes that decay into stable daughter isotopes at a perfectly constant rate. The half-life is just the time it takes for half the parent stuff to decay.
Like a very, very slow hourglass.
A perfect analogy. A natural hourglass that never changes speed. So something like carbon-14 has a half-life of about 5,700 years. Good for dating relatively recent things, maybe up to 75,000 years old.
Okay.
But for really old rocks, we use isotopes with much longer half-lives. Uranium-238, for instance, its half-life is 4.5 billion years.
Wow. Nearly the age of the Earth.
Right. And since you usually can't date the sedimentary rock the fossil is in, you date layers of volcanic rock-like ash falls that are above and below the fossil layer. Those volcanic rocks trap the right isotopes when they cool.
So you bracket the fossil's age. That's clever.
It works remarkably well.
And when you put all these dates and layers together, that gives us the geologic record, right? Earth's official timeline, divided into eons and eras.
Exactly. 4 eons: Hadean, Archean, Proterozoic, Phanerozoic. And the Phanerozoic, the last half-billion years or so, is split into eras: Paleozoic, Mesozoic, Cenozoic.
And the boundaries between those eras, they often line up with huge extinction events, don't they? Like massive punctuation marks in Earth's history.
They absolutely do. It's pretty humbling when you think about the sheer scale of time involved.
Right.
Billions of years.
Our entire human history is just a tiny blip at the end.
A blip is right. Let's try that 1-hour clock analogy. Earth's 4.6 billion years is 1 hour. Prokaryotes, the first life. They show up really early, maybe around the 10-minute mark. forming those layered rocks called stromatolites about 3.5 billion years ago.
Okay.
Then atmospheric oxygen starts to rise significantly around 2.7 billion years ago, thanks to photosynthesis. That's a huge change. Eukaryote cells with a nucleus, like ours, maybe around the 38-minute mark, 1.8 billion years ago. Multicellular life, maybe 1.3 billion years ago, so around 45 minutes in.
Still no animals yet.
Nope. Animals only appear around the 51-minute mark, maybe 9 minutes before the hour's up. And humans, we appear in the last 0.2 seconds of that hour.
0.2 seconds.
Yeah.
Wow. That really does put things in perspective.
That's it.
And this timeline lets us trace how whole new groups evolved, like mammals. How did we get here? Can you walk us through that? The origin of mammals seems like a great example of macroevolution.
It's a fantastic one. Mammals have these distinct features, things that fossilize well, like the single bone in the lower jaw, that specific jaw hinge, the 3 little bones in the middle ear, different kinds of teeth.
Right, incisors, canines, molars.
Exactly. And the fossil record shows these features didn't just pop up overnight. They evolved bit by bit over something like 120 million years, starting from a group of ancient 4-legged animals called synapsids.
120 million years.
Yeah.
That's a long tinker. And wasn't there something about jaw bones becoming ear bones? That sounds wild.
It is wild, but it's a classic example of how evolution repurposes things. So in those early synapsids, the jaw joint involved 2 specific bones. But over time, the main lower jawbone got bigger and formed a new, stronger joint. Those 2 original hinge bones, they weren't really needed for chewing anymore.
So they just vanished.
Nope. They shrunk, migrated inwards, and became the malleus and the hammer and anvil bones in the mammalian middle ear. They were repurposed for transmitting sound.
No way. Jaw bones became ear bones. Evolution is like the ultimate recycler.
It absolutely is, tinkering with existing parts to create something new.
Okay, so we see life changing over these vast timescales. But what's actually driving these big changes? What are the engines of macroevolution? Because it feels like a balance, right, between new species appearing speciation and species disappearing extinction.
That's the core dynamic. And one of the biggest drivers operating on a planetary scale is plate tectonics, continental drift.
The continents moving around.
Yeah, Earth's crust is broken into these huge plates floating on the mantle underneath. They move slowly, just a few centimeters a year, but over millions of years.
It adds up. How do we know how they moved in the past?
Geologists track it using magnetic signals locked into rocks when they formed. It gives us a map of how continents have split apart and crashed together over deep time.
And the consequences of continental drift for life must have been massive.
Oh, dramatic. Think about when the supercontinent Pangaea formed about 250 million years ago. It smashed continents together.
What did that do to life?
Well, it destroyed a lot of shallow coastal seas where most marine life lived. And the inside of that giant continent became super dry and harsh, lots of extinction. But it also spurred allopatric speciation on a huge scale.
Meaning species got separated and evolved differently.
Exactly. As continents later broke apart, populations got isolated. Think about, say, related frog species found today in Madagascar and India. They diverged because those land masses split apart millions of years ago. It also explains why we find identical fossils, like certain ancient reptiles, in both Brazil and West Africa. They used to be connected.
Right, okay. So continental drift drives both extinction and speciation. What about those really big extinction events you mentioned, the mass extinctions?
Yes, the Big Five. These are times when a huge percentage of species worldwide disappear in a, geologically speaking, short time. They're major turning points.
And the 2 most famous are the Permian and the Cretaceous, the dinosaur killer.
Those are the best studied, yeah. The Permian mass extinction around 252 million years ago was the big one, absolutely devastating. Maybe 96% of marine species wiped out.
96%. What could possibly cause that?
The prime suspect is enormous volcanic eruptions in what's now Siberia. Just unimaginable amounts of lava releasing massive amounts of CO2.
Leading to global warming.
Extreme global warming, yes. Plus ocean acidification, which made it hard for creatures to build shells. And widespread ocean deoxygenation, maybe even poisonous hydrogen sulfide gas bubbling up, a real planetary catastrophe.
Okay, that sounds grim. Then there's the Cretaceous mass extinction, 66 million years ago. That's the one that got the dinosaurs, right? Except birds.
That's the one. And the big clue there is a thin layer of iridium found all over the world in rocks from that exact time.
Iridium. That's rare on Earth, isn't it? But common in asteroids.
Bingo. That was the smoking gun pointing to a massive asteroid impact. We even found the crater Chicxulub. off the coast of Mexico.
So asteroid hits, massive debris cloud blocks the sun.
Exactly. Global cooling, darkness, collapse of food chains. Bad news for dinosaurs, but maybe good news for small furry mammals like our ancestors.
Which raises a kind of uncomfortable question for you, the listener.
Yeah.
Given what's happening today, are we in a sixth mass extinction?
It's a very serious question scientists are grappling with. Current extinction rates are estimated to be, well, maybe 100 to 1,000 times higher than the background rate we see in the fossil record.
Driven by us, habitat loss, climate change.
Largely, yes. Human activities are the main drivers. Now, have we reached the scale of the Big Five in terms of percentage loss? Not yet. But the rate of decline is deeply alarming. It's a stark warning.
And the consequences of mass extinctions, even if we're not fully in one yet, are huge. They don't just reduce numbers, they wipe out entire evolutionary lines forever.
Gone for good. And recovery takes an incredibly long time. Millions, sometimes tens or even hundreds of millions of years, like after the Permian, ecosystems get totally restructured. Sometimes predators become much more common afterwards, for example.
But there's an upside, sort of, a flip side to extinction.
There is. It's called adaptive radiation. After a mass extinction clears the board, or when organisms reach a new place with lots of opportunities, or even develop a major new evolutionary trait.
They diversify rapidly, fill the empty roles.
Exactly. Periods of really rapid evolutionary change, new species forming quickly to exploit those vacant ecological niches.
The classic example is mammals after the dinosaurs vanish.
Perfect example. Dinosaurs disappear, and suddenly there's all this ecological space. Mammals, which had been mostly small and nocturnal, radiated out into an amazing diversity of forms and sizes.
Filling all those roles the dinosaurs left behind.
Right. Or think regionally, like the Hawaiian Islands. You get the Silver Sword Alliance, descendants of one ancestral plant that arrived maybe 5 million years ago, diversified into dozens of unique species adapted to all the different island habitats, from wet forests to dry volcanic slopes. It's stunning.
Okay, so we've seen the what and when, fossils, timelines, extinctions, radiations. Now, how does this happen at the genetic level? How do small changes create these big shifts in form?
Now we're getting into evo-devo evolutionary developmental biology. This field looks at how changes in the genes that control development, how an organism grows from an embryo, can lead to major evolutionary changes in body shape and form.
So changes in how creatures develop.
Precisely. One key mechanism is heterochrony. That just means changes in the rate or timing of developmental events.
How does that work? Give me an example.
Okay, think about human versus chimpanzee skulls. A lot of the difference comes down to different growth rates of various skull parts, especially the jaw during development, or bat wings.
Bat wings.
Yeah, a bat wing is basically a modified mammalian forelimb, but the finger bones grew much, much faster and longer due to changes in developmental timing.
Wow. And the opposite can happen too, like whales losing their hind limbs.
Exactly. In whale evolution, the growth of the hind limb buds slowed down drastically during embryonic development, eventually leading to their near-complete loss in modern whales. Small tweaks in timing, big results.
So timing is key. What else?
Well, there's paedomorphosis. It's a type of heterochrony where the adults of a species retain features that were juvenile in their ancestors.
Like they never fully grow up in some ways?
Kind of. The classic example is the axolotl, that Mexican salamander. Most salamanders lose their gills and become terrestrial as adults. But axolotls become sexually mature while still keeping their gills and aquatic lifestyle, like a permanent larva.
So a change in development keeps them in the water.
Right. Again, relatively small genetic changes impacting development can lead to really different looking and behaving animals.
Okay, so changes in timing. What about changes in where body parts develop? Like how does an embryo know where to put the legs or the head?
That's governed by changes in spatial pattern, controlled by master regulatory genes called homeotic genes, especially a famous set called Hox genes.
Hox genes. I've heard of those. They're like the body plan architects.
That's a great way to put it. They provide positional information. They tell blocks of cells, you're in the head region, develop head structures, or you're in the thorax, develop legs here, or not.
Or not. How does that work?
Look at insects versus crustaceans like shrimp or lobsters. Insects famously have 6 legs all attached to the thorax. Crustaceans often have legs on their abdominal segments too.
Right.
Research shows that in insects, a specific Hox gene is active in the abdomen, and where it's active, it suppresses leg development. In crustaceans, that suppression doesn't happen the same way, and scientists have even found specific changes in the protein sequence of that Hox gene that seem linked to this difference.
So a gene that says don't build legs here is key to the insect body plan.
Mm,
Fascinating.
yeah. It really is. And this leads to another crucial insight from Evo Devo. Sometimes, changes in gene regulation are more important than changes in the genes themselves.
Meaning when and where a gene is turned on or off, rather than changing the gene's actual code.
Exactly. Think about it: if you change the protein a gene makes, that protein might do many jobs in different parts of the body. Changing it could cause problems everywhere, but changing where or when it's turned on, that can have a more localized effect, maybe with fewer negative side effects.
Like using a dimmer switch instead of replacing the whole light bulb.
Nice analogy. And we see this beautifully in three-spine stickleback fish.
Sticklebacks, those little spiny fish.
Yep. Marine sticklebacks usually have these prominent spines on their underside, probably for defense. But many populations that got isolated in freshwater lakes, where maybe a big predatory fish are absent, have lost those spines.
Okay, so they evolved to lose the spines. Did the spine-building gene break?
That's what you might think. But researchers found the gene itself, called PIX1, was perfectly fine in the spineless fish. The change wasn't in the gene sequence. It was in a nearby region of DNA, a non-coding bit called an enhancer, which acts like a switch to turn the gene on in the specific area where the spines would develop.
So the switch broke, not the gene.
Essentially, yes. A
Yeah.
change in regulation meant the gene wasn't turned on in the pelvic region, so no spines developed there, even though the gene worked fine elsewhere in the body. It's a beautiful example of how evolution can target changes to specific body parts through gene regulation.
Okay, this Evo-Devo stuff really changes how you think about evolution.
Yeah.
It's not just random mutations, it's changes in this underlying developmental toolkit.
Precisely. And it reinforces the idea of evolution as tinkering. It's not designing things perfectly from scratch. It's modifying what's already there, tweaking developmental pathways, repurposing old structures,
Like the jaw bones becoming ear bones.
or complex structures like the eye. The vertebrate eye is incredibly complex, but it didn't appear fully formed. It evolved step by step from simpler light-sensitive patches, like you see in limpets today.
Just detecting light versus dark.
Right. And each intermediate step, a slightly better light-detecting organ, provided some advantage. What's amazing is that complex, image-forming eyes evolved independently multiple times in different groups. Vertebrates, mollusks like squid and octopuses.
Using different internal wiring, so to speak.
Different structures, yes, but all building upon the same fundamental ability of cells to detect light. Tinkering, again.
And this tinkering leads to exaptations.
Right.
Structures evolve for one thing, get co-opted for another.
Exactly. The ear bones are the prime example we keep coming back to. They evolved from jaw bones. Bird feathers might be another, initially evolved perhaps for insulation or display, only later co-opted for flight. Natural selection can only work on a structure based on its current function. It can't plan for a future use.
It's just what works now.
What works now, incrementally?
This also helps us understand evolutionary trends, doesn't it? Like the classic story of horse evolution.
Ah, yes, horses. From little dog-sized Hyracotherium with multiple toes browsing in forests 55 million years ago to big single-toed equus grazing on grasslands today.
When I first learned that, I pictured this neat, straight line. Like evolution was aiming for the modern horse.
That's the popular image, but it's misleading. If you look at the actual fossil record, the horse family tree is more like a dense, bushy shrub than a straight ladder.
Lots of dead ends and side branches.
Exactly. Equus is just one surviving twig on that bush. There were many other kinds of horses, some big, some small, some with different numbers of toes, that lived alongside each other and eventually went extinct. There wasn't some internal drive towards equus.
So the trend towards larger size and fewer toes wasn't inevitable.
Not inevitable, no. But trends can still be real patterns over time. They might result from species selection. Maybe lineages that speciate more often or last longer tend to have certain traits, influencing the overall pattern. Or it could be driven by consistent natural selection in a changing environment.
Like, as grasslands spread, horses better adapted for grazing and running on open ground were consistently favored.
That's likely a big part of the horse story. But the key point is, evolution responds to current conditions. If the environment changed again, that trend could slow down, stop, or even reverse. Evolution has no long-term goals.
Okay, so wrapping this all up, what are the big takeaways from this deep dive into macroevolution?
Well, I think we've seen how vital the fossil record is, even with its gaps, as our guide to life's immense history.
And how huge geological forces like plate tectonics moving continents around and catastrophic mass extinctions have profoundly shaped the trajectory of life.
Absolutely. They're major drivers of large-scale change. And then we saw how the importance of developmental genes, the Evo-Devo perspective, how small changes in timing, location, or regulation of gene activity can lead to really major changes in form. Evolution as tinkering.
It paints a picture of life as this incredibly dynamic branching process, constantly responding to changing conditions, punctuated by these big events.
It really does. And it may be a final thought for you, the listener, to ponder. Think about the contingency, the sheer chance involved. Consider mass extinctions again.
Like the asteroid 66 million years ago.
Exactly. If that asteroid had missed, or if the early primate lineage, our ancestors, hadn't survived that event, humans simply wouldn't be here. Life on Earth would look profoundly different today.
It really highlights how unpredictable the path of evolution is. We're here because of this long, intricate, and often chancy history of tinkering and survival.
Precisely. Our existence is a product of that deep, winding history.
Well, this has been absolutely fascinating. A huge thank you for joining us on this deep dive into the broad patterns of evolution. We really hope you found it insightful.